Hi, you're watching DNA Today, a multi-award winning genetics podcast where we explore everything to do with genetics, from CRISPR to rare diseases to new research. We have won the Science and Medicine Podcast Award for many years now. We have hundreds of episodes, and we really hope you enjoy these conversations where we dive into so many genetic concepts. I'm Kira Deneen, a certified genetic counselor, and your host.
Hi, everybody. I am very excited for this episode. I have waited literal years to cover this topic on, I feel like we should call it RNA today, which I have to say, that was Rachid's comments of what we should be doing, our guest today. So we're going to be exploring the role of RNA analysis in genetic testing. And again, I've wanted to do this episode for a long time, but I was holding out for Ambry Genetics because, in my opinion, Ambry Genetics is the leader in the space when it comes to RNA, and we're going to break down why that is and how it's so helpful. So as I mentioned, Dr. Rachid Karam is the Vice President of Research and Development at Ambry and their head of Translational Genomics Lab. And we're going to be talking about Ambry Genetics' history of innovation in RNA testing, how it is increasing diagnostic yield and reducing variants of uncertain significance in hereditary cancer testing, and its role in closing health disparity gaps. We're also going to introduce a brand new test from Ambry called ExomeReveal, and this includes RNA analysis within exomes to be extending our expertise in rare diseases. So, Rachid, we have a lot to cover, don't we?
We do. Yeah. Thank you so much for having me, Kira. I love your show, and even though I'm the RNA guy, I do watch DNA Today, listen to DNA Today pretty frequently. So
I love that. Yeah, I feel like today is RNA Today, so kind of interesting spin on things. And as I said, I see Ambry as the leader in RNA analysis. This has been on my list of kind of dream episodes to do. So it's really a treat to be able to record with you today. I want to start by saying and understanding a little bit more from you of why Ambry is the leader in RNA analysis and how Ambry became a pioneer in RNA testing, and what were some of those initial challenges and breakthroughs for Ambry to get to the point of even offering this as part of testing.
Ambry has a long history of firsts, particularly right for rare disease. One of the first to launch exome testing. I think even panel testing, which today it's like gold standard. If you think it wasn't the case last decade, right? Like a lot of testing was done like single gene testing in the cascade fashion. So there was a shift in mentality, and I think Ambry was one of the key players to help implement this mentality shift that look at the realities of our health system, right? How they behave, pretty much the test once opportunity kind of scenario. So let's leverage this. And Ambry has always had that mentality with the multi-gene panel testing. Let's test with new technology, pretty much embracing new technology early on and taking that to the benefit of the patients, right? And I think there was a point during Ambry is celebrating its 25th anniversary this year, but so we have seen the transition from Sanger to NGS, and that was a huge transition because it allowed us to go from the single gene testing to now much bigger panels, right? And in adopting that technology, NGS early on was key. And I think the same mentality with RNA. I think as soon as we realize that there was that was important for the patients, we start investigating and then eventually bringing that to the clinical arena.
And how does RNA testing actually work? Because obviously we talk about DNA testing a lot on the show, but we haven't really explored like, well, how does it actually work? And that might be helpful for people to understand as we get into how it's actually helping with the data we're getting in the end and closing that diagnostic odyssey in different aspects there. So how does it work, and how does it help with classifying variants more accurately? Because I think that will kind of tie in together.
Yes. If you think too, like with the advent of multi-gene panel testing, right? So NGS, that was a really cool technology. Now you can add as many genes as you want to a panel. What was the caveat of it? Is that now we were getting data from genes, from variants that we didn't understand very well, right? So we saw as multi-gene panel testing got implemented, we did see a rise on variants of uncertain significance, VUSs. And that became like a really big problem for the industry, right? So like imagine you're a clinician, you ordering a test and very often the result comes back inconclusive, uncertain. We don't know what that variant that we identify on the DNA testing does, right? What does that mean? And I think that's challenging for clinicians. It's even more challenging for patients. Can imagine having to explain to them what a VUS is. And so having that understanding that it is a big issue for the industry, right? Variants of uncertain significance, period. And how can we deal with them? How we can solve them? And RNA turned out to be the low hanging fruit, pretty much, because we knew that a lot of those VUSs, particularly for those in cases where the phenotype was aligned, so we had a lot of confidence that some of those variants were causative, right? They were associated with the disease, but we did lack the evidence to show that, right? And RNA provides that evidence. So you looking at the expression part, you're looking at the splicing level, and you can see then the functional impact that a DNA variant has on the mRNA. And if you think, splicing is really fascinating, right? Like the cell can tell with one nucleotide precision what's a coding sequence, what's an exon, and what's encoding, what's an intron, and then go there and remove those huge intronic areas of the gene and come up with like a messenger RNA that that codes for a protein, right? And does that with such a level of precision that I find that it's fascinating. And to being able to predict what the variant does, it's a tough business, right? So if you only looking at the DNA, if you're only looking at in silico tools, it's hard sometimes to be able to tell what the outcome is going to be molecularly. And that's where RNA makes all the difference, because you actually have the data handy, right? It gives you more data, and more data, it's everything when it comes to variant interpretation. And we see that impact when we are analyzing variants that were previously VUS. After we add the RNA data, following, of course, all the American College of Medical Genetics recommendations, right? We apply that evidence, we can shift a good proportion of those variants from VUS into either clinically actionable, or if that's the case, right? Or if there is no impact on the splicing, several of them we can downgrade as likely benign, so not associated with disease.
It's an important tool to have to be able to figure out, okay, this variant that we found, is that going to lead basically the RNA to not be produced correctly, or is it not? So it's surprising to me that in 2024, when we're recording this, not all labs are really taking advantage of this tool that can be a game changer in terms of, is it a VUS? Oh, wait, we have this extra information. Let's apply that. Now maybe we can, as you said, like downgrade it to, one of the other variants, likely benign or benign. I think that's really important, especially when it then differs on clinically what's happening. So I think when it comes from that perspective, it's really important. But, as I was saying, like other labs, they may offer RNA testing in some capacities, but labs may decide when to leverage this technology, and it often isn't performed concurrently with the DNA analysis. And from my limited understanding, it seems like with Ambry, that's what's happening. You guys are doing it either at the same time, or you have it done before the report is given. At least in the cancer space, that seems to be the case of how you guys are operating. Right. Why does Ambry feel that this is the best approach? Am I getting it right in terms of like having it all at once is really helpful?
So this got started with cancer, right? And remember, back when we launched this product before in 2019, before the RNA test was available, we did a lot of translational research. So Ambry has our Ambry Translational Research Lab, and that's a lot of where this technology was initially evaluated. And we started on a safe space, if you will, in terms of like cancer, there are a limited amount of genes that we know that are associated with the phenotypes, right? And these are genes that we understand well. So it was a huge challenge itself just to implement something new clinically, period. And I think we were well positioned. I think why Ambry? I think we were well positioned because we done so before we transitioned from Sanger to NGS to panel testing. We done before as well too with CNV calling. There was a time people that are new to the field may not even remember that like del-dup analysis, like the CNVs, wasn't even part of the clinical testing. You would do the gene sequencing, and if that was negative, you could cascade to do the del-dup analysis later on. Ambry we added that to the panel, right? So I think that mentality, and that's back to that, our health system, how it works. Many people, they only have one chance to get those type of tests performed, right? So I think really like to make this work, I think the idea is let's offer to the patients as much as possible in terms of molecular tools to get us the best result possible. So increase the positive yield to decrease the VUS rate. All this data matters. And we start that with cancer because those were genes that we understood better. And it was much easier to implement that clinically at that scale. And I think that has helped initially the intention to reduce VUS, which we did see a 70% reduction in splicing VUSs. So and a lot of those VUSs turned out to be clinically actionable. So those were patients that were not getting the treatments that they needed or preventive risk reducing procedures, right? Just because they lacked the classification, right? So we did see a lot of those variants being reclassified on cancer. And also one thing that the cancer test allowed us to do is to start detecting those deep intronic variants. So those were variants that were not necessarily easy to detect at the DNA level. And a lot of missing heritability turns out to be explained by the deep intronic variants. I remember you have a podcast about FAP. I think that's a really interesting phenotype, the polyposis phenotype, right? And we have a paper that we just published at GCO Precision Oncology where we show a bunch of those families that clinically they had polyposis, they had FAP. The clinicians were like, how is it that this individual is negative, even though after done DNA testing? We did RNA, we found deep intronic variants, right? So like those were variants that we couldn't detect before. So that was kind of like a bonus we got from RNA in cancer. So not only reducing the VUS, but also detecting variant types like deep intronic and other structural variants that you cannot really see them at the DNA level as well. And now I think it's time for rare disease. All this that we learn from cancer, I think it's the time for us to start applying this type of technology. We learned its limitations. We learned a lot about like how implementing this at scale, right? And now I think the time has come for us to finally start to tackle the VUSs first on the exome on rare disease, right? And then eventually, this is just going to be the first version of the product, of course, then eventually getting all the other benefits you can get out of RNA. I think rare disease has a ton of potential because even more potential than cancer, because we're talking about the entire genome, right?
Yeah, I think that makes sense in terms of taking what Ambry has learned in the hereditary cancer space and finding those genetic changes, being able to use RNA to give more information to then classify those variants, taking that same workflow and applying that to exomes in like for rare diseases and undiagnosed, conditions, obviously at that point. So I think that it seems like a natural next step to be applying this technology. And before I get more into asking about, rare diseases and using RNA with exomes, I was just thinking of, will we get to a point where we could just analyze the RNA and we wouldn't need to look at DNA and RNA? Like, am I going to have to rename this to RNA today, like in the future? Are we just going to be focused on RNA? Is DNA kind of going to be like, oh, back in the day, we looked at people's DNA? Or are we always going to have both?
I think they complement. I don't like to speak in terms of like always. I don't know, to be honest. But I think they complement each other. I think our reports, they always going to be DNA based at ours for now, right? Because it's really like that's the variant that the counselors are going to use the clinicians going to use to dictate what are the next steps, the DNA. But then the RNA data, I think, and overall, we're really at the infancy of this business. If you think like where radiology was 100 years ago and how much has it progressed, right? This is the tip of the iceberg. I think we are very eager to make progress, but we're just getting started. And I think this is one example, right? Like for a long time, genetic testing was synonymous of DNA testing, and not anymore. Right, and I think there are so many more things that we still can keep adding to testing, methylation, proteomics, right? And I think the more the better in the sense. And I think there is a ton of things from RNA that we haven't explored yet. For example, expression profiles. I know there are other labs that do diagnosis, for example, to see people that are responder or not to rheumatoid arthritis medication, right? So based on their RNA profile, they can tell whether that person is predicted or not to respond to specific medication and then tailor their treatment on that. I think PRS, polygenic risk score, it's one thing at the DNA level. I think we need to start thinking at the RNA level, how does it impact expression, right? And then that ultimate question, why people with the same variant, you may have let me use my favorite gene, CDH1, and have one specific phenotype, someone with hereditary diffuse gastric cancer, someone with lobular breast cancer, and another individual with like a cleft lip, right? And the same variant. And there's a lot of expression and a lot of things that we still need to learn. So I think this is the beginning, and we are excited with the things that are to come as well.
Yeah, there's so much to learn within it, and genetics is just so complicated. That's how we're able to have a weekly genetics show, right? Because there's so many topics to explore, and even within each topic is so complicated, right? So I think it's just so helpful to be able to have RNA testing to complement all of this, to figure out, especially we've had so many episodes about different rare diseases and how, a lot of the times we're talking about kids with rare diseases, and oftentimes the kids will have the same exact genetic change that we think is causing those symptoms, but their symptoms are a little bit different, or what age those symptoms come on is a little bit different, or the severity of that. And so we know that there has to be other factors at play that are tinkering in terms of those aspects. And I would imagine looking at RNA is going to help us figure it out. And even certain conditions, we did an episode about like a genetic cause of epilepsy, end of July, early August, somewhere in there. And it's just interesting because a lot of those kids, they're kind of like two different phenotypes, but it's the same either genetic change or the same gene. And they don't find that, oh, this genetic change is this phenotype. So there's so much to explore within this that is really going to affect not only the diagnostic odyssey and getting an answer for people, but also to know more details within that and to affect the kind of treatment they might have and what's going to be helpful. And something else that I learned about with your colleagues back on episode 297 is that using certain types of testing and making sure that those databases are really, really have a variety of different patients within them in terms of ancestry, that can be very helpful. And what I'm learning too is that RNA testing can also benefit in that same way of being able to help to classify variants, because there are certain ethnicities such as European descent, white people, that we have a lot of information on. And there are other ethnicities and where people are from in other areas of the world where we don't have as much information on. And that tends to be more VUSs in that area. So I'm imagining RNA testing is really going to help with that, not only in the cancer space, but in this rare disease space as well.
Absolutely, 100%. And I agree with you. It's a very complex business, right? With all the new advances, we tend to think like it's simpler. but it's still very complex. And I think particularly data matters. So more data, it's better. Expertise matters, right? So who is doing the interpretation of the data? And particularly when it comes to the gaps that we see, particularly in terms of variant assessment, a lot of the ethnical backgrounds, we just don't have as much data available for them. Period, right? And so RNA, it's really an opportunity to getting that sample being collected. You will get that data from that individual, and having it does make a difference. We have data published on the cancers, and but also I'm quite confident a lot of those things that apply to cancer, they apply to rare disease as well. As long as the gene is loss of function, right? As long as the gene has introns and behaves with splicing and all that, right? Those are rules that are molecular rules, right? So yeah, things that apply to CDH1 apply to BRCA1, apply to ATM. They will apply to other genes that cause rare disease as well too. So being able to leverage that data, it definitely, the same way it did have impact on reducing VUS rates for non-whites in cancer, I'm quite confident we'll have a similar impact on rare disease. And the same thing, but overall, I think every patient will see a benefit, right? Like less VUS is more positives, but particularly subgroups that have lacked in data with regards to data accessibility. I think this helps by providing another data point that we can leverage.
Yeah, that's so important to be able to have that, because that's notoriously been an area of genetics that we have really needed to improve. And I think, you have more experience than I do of just being around in the field longer, but in the sense of I think there's been more of a focus in the last 5, maybe 10 years of really expanding these databases to reflect the human population. And it's something that we need to keep chipping away at. And I feel like this is a really big piece to that, because it almost makes it like personalized medicine in that way, or personalized diagnosis, I guess, but within that area of precision of looking at that person and saying, okay, well, what do we see? How is this DNA actually being expressed? It's that next level of it. And one thing that you said was looking at loss of function. So that makes sense in terms of that mRNA is not being produced at a high enough level. What about the opposite, gain of function? Are you able to look at, how much RNA is being produced, or is it more like it's there or not and we're sequencing it?
Yeah, the test that we perform, it is quantitative and qualitative, right? So as you mentioned, it's important when you're looking at a splice variant, does it result in an mRNA, in a transcript that is out of frame, right? That's one thing that it's qualitative on that sense, that tells you specific. So if it is out of frame, it's going to result in a premature termination codon. The RNA is unstable, maybe degraded by nonsense mediated decay. So you don't even have RNA or protein in the end, right? So that would be the loss of function path. For gain of function, it's a little bit more complicated. A lot of the germline cancer genes, most of them are loss of function. And usually those are the ones that we are applying RNA evidence now, specifically for splicing variant classification. If it is gain of function, usually we can apply RNA for splicing, right? But thinking about the future, I think that as part of expression profiles and things like that, we know when we're looking not at germline, but at tumors, yes, RNA, it's important in gain of function to detect fusions that may be being overly expressed and things, and other examples as well too. But usually for rare disease and hereditary cancer, we're more concerned. RNA is going to be more beneficial to detect those loss of function transcripts, right? And that's why it's so straightforward. It's almost like the same. You have the variant at the DNA level, and now you have the RNA. You're showing that premature termination codon in the RNA, so that's not going to lead to a functional protein. And that's why we can apply so much weight to that evidence when we class.
Yeah, that's really helpful to be able to have that and know that it's helpful in both situations. A little harder in one, but that it's valuable information to help you, figure out that result and what's going to be information that's going to help in terms of the clinical treatment or that diagnosis. So another thing that I was just thinking about is that you mentioned the diagnostic yield is much better in the hereditary cancer like panels and looking at that. I know ExomeReveal is new, so I don't know how much information you have at this point, but what do you foresee in terms of that diagnostic yield using RNA with exome in that space? Because I would think that it's also going to be higher.
Absolutely. All the preliminary data that we have shows that. In a way, now, as you mentioned, we do have a new test, ExomeReveal, that is pretty much incorporating RNA testing to exome, right? But it's a new clinical test. We have been doing translational research on the back end for all these years to try to understand what impact RNA may have for rare disease. And we presented at the ACMG in Toronto this year some interesting data for neurodevelopmental disorders. So where we looked at a series of splicing variants that were detected in phenotypes other than cancer neurodevelopmental in this specific case. And part of that work, number one, we were like, how many genes actually can we look at based on blood, right? Because remember, it's a blood test. And then there is always that issue. Is this gene expressed in blood, number one? And is this expression in blood similar to what we would detect on the tissue of interest or the developmental stage that this may be important? So we did show at that ACMG data that about 80% of the neurodevelopmental genes that we looked, blood would be a good surrogate. So we would be able to use blood to do splicing analysis for those genes. So about 80% of the genes that we looked. And then what we observed is that if the variant it's affecting, predicted to affect splicing, or if it's an intronic variant, we were able to reclassify up to 90% of those cases. So it's really effective in the sense that if it is the right type of variant, if the gene is properly expressed, we will probably be able to give the patients an answer. And that was the neurodevelopmental field, particularly for exome, as we prepared to launch the ExomeReveal clinically. We did have an early access phase where we were trying to understand about what proportion of the cases would be resulting in RNA testing. We're seeing about 5% of the exome tests benefiting from directly from RNA, right? And yes, we already have examples from this early access phase of clinical cases that have been identified that would have been classified as VUS if it was DNA alone. But because we had the RNA sample, we were able to give them an answer. And the one main difference, remember when we were doing this as translational research, we were talking about years. I used to say it's research turnaround time, right? This is research, it may take a long time. The major difference from the clinical test is that now there's a turnaround time, weeks. And so it really speeds this process. And I find it's amazing. There was a point where we were even wondering, are we going to ever be able to do RNA in rare disease, or we will need like IPS differentiated into brain organoids to be able to get meaningful data out of those individuals. But no, RNA, we can get good RNA from blood as long as it's collected on the proper tube, the PAX tube. The quality is great and allows us to do a lot of things, splicing being one of them. And we definitely already seen patients benefiting from this technology.
Wow, that is remarkable. And I'm just thinking of like how this is going to be a game changer in the field. We've already seen it in cancer and that it has completely changed how we're able to do things. And now to have this in the rare disease area, in the pediatric realm, like that is just remarkable. I think people are going to look back and be like, wow, remember when that was launched? And it's going to be a big milestone, I think, in genetics. And one other thing I was thinking about it as we kind of start wrapping up is that it's been a while since I've been in pediatrics. And when I was a student, right? So I don't know as much as I do in prenatal. But we would do, when we were ordering exomes, we would order as a trio. So we'd have the child and then we'd have both biological parents. And that is to kind of figure out, well, if we find a variant and we think, oh, well, maybe this is it. If we look at one of the parents and they have it and they're healthy and they don't have the symptoms the kid does, oftentimes we say, well, that's probably not it. And we keep looking. Do you see that using RNA with exome would be a way of replacing the trio testing? Because both biological parents are not always available for that test. Or is it really two different things?
I think there are two different things. I don't think we'll be able to replace Cure because again, the trio provides more data that we can use for other things like de novo information, right? Which turns out to be very common for rare disease. So there are things that we learn from that. And that's part of like getting more data. I think RNA will complement that. Will be more that we can add. In terms of future directions, I think things that RNA could be a game changer. So remember, this is the first version. It's really for rare disease. It's really focused on VUS reduction, which sometimes it's a topic for rare disease, I believe, as well too, because a lot of tests don't even report VUS because otherwise there will be such a ginormous list. So a lot of times we put this dust under the rug, right? I don't even want to look at those VUS because there's nothing I can do with them. Now I think that's really, let's reevaluate that conversation because there may be VUSs that may be explaining phenotype these people were previously reported as negative. I think that's why our Patient for Life program, it's important. It's really, there is now new data that has been identified. And yeah, your patient was negative 5 years ago, but now the gene, it has been identified as associated with disease. And now your patient has a variant of that gene. And if it's a splicing variant, we now have the tools to classify that variant properly too. So I think the first step is going to be really VUS reduction. And then we'll be able to leverage this technology as well with as other tools to get.
I think combining the Patient for Life program, as you're mentioning, we explored that with your colleagues in episode 297 of revisiting the exome, and Ambry's doing that just, on your own. And then you let the clinicians know, hey, this variant's now been updated. That's just so helpful. And I'm not going to get the quote right, but Liz had a great quote basically saying that the exome that we have today is better than yesterday, but not as good as tomorrow. And I think once you put in now this RNA analysis level to it, it's just going to be that much more powerful that as time goes on, just like you were talking about, we went from Sanger sequencing to next generation sequencing. We're going to see that in certain cases, we're not ordering panels as much. We're ordering more exomes. There's always going to be a time and place, I think, for panels. But I think especially in this rare disease area, looking at exomes is going to be more and more ubiquitous in terms of when we have just those generic findings of global developmental delay, seizures, and it's just those that we're like, well, we don't know where to start. That often, is, I think, going to become even more common to have exome be the first test that's ordered. But thank you so much, Rachid. This was just so interesting. And as I said, I've been really wanting to do this episode for a long time. So it was just so interesting to dive into all of this with you. And I know we could just keep talking forever, but anything you want to leave our listeners with today?
Just thank you. This was a great opportunity and I'm excited. I'm excited to have RNA finally impacting the patients with rare disease. I think that's looking forward to see once this gets launched clinically, that's when you start being able to look at things real life, right? And so I'm looking forward to see how that's going to look.
As am I. We might have to do a follow-up of like, okay, it's been X amount of time. How has it gone? But thank you so much, Rachid. This has been really fun. I really appreciate it.
My pleasure. Thank you.
Thanks for watching DNA Today. To access all of our episodes, head over to dnatoday.com. We also have a lot of bonus content on there that you can enjoy. If you have any questions, comments, suggestions, guest pitches, you name it, send them in to info@dnatoday.com. We'd also really appreciate if you could take a moment to rate and review the podcast on Apple, Spotify, wherever you listen to the show. It really helps more nerds like yourself find the show. Also, if you like giveaways and other ways to connect with us, I recommend following us on social. We're at DNA Today Podcast. We also have a Patreon if you want to be the most level involved in the show. That's also at DNA Today Podcast. Thank you so much for listening and watching. Join us next time to learn and discover new advances in in the world of genetics.